Well, welcome to this third part of the iBio seminar on rafts in cell membranes. I'm Satyajit from the National Center for Biological Science, working in close collaboration with my physicist colleague, Madan Rao, trying to make sense of what we see in living cell membranes. So if you remember, or in case you haven't seen the previous part, what we had observed was that many different lipid-anchored proteins are organized as small nanoclusters in membranes of living cells in a manner that seems consistent with their being formed by some active process because they exhibit a concentration independent clustering phenomena in these in cells and living cell membranes. We had also proposed that these nanoclusters of lipid-anchored molecules may come together and form regions in cell membranes that we believe are functional domains which can perform sorting and signaling functions under regulatory control of the cell's machinery. Well, the questions that we were left with at the end of the last part were questions about what makes these nanoclusters of these different lipid-anchored molecules in cell membranes and what induces their forming functional domains. And I think if we can address those two issues, we would be well on our way to understanding functional domains in membranes of living cells. As you will appreciate, coming to this point has required a development of new technology, new tools to image processes in living cells at the scales at which they demand. We've had to develop new types of microscopy to be able to study and elucidate the structure of these little nanoclusters in living cell membranes. And in fact, understanding and beginning to observe the formation of these functional larger scale domains also has required new developments in microscopy. So what we have been able to do is image the signals that we had observed from these nanoscale clusters, the FRET signals, the fluorescence or Foster's resonance energy transfer signals from these nanoscale structures. We've begun to image them at much higher resolution using a high resolution FRET imaging technique where we are able to map the FRET using this HOMO-FRET technique that we had developed in the previous part, in the previous iBio seminar segment, at much higher spatial resolution. So the spatial resolution that we have now achieved is about 300 nanometer resolution. And at this resolution, if we begin to image the FRET signals coming from these lipid-anchored proteins, specifically GPI-linked proteins at the surface of living cells, we see the following. So in living cell membranes, here is the fluorescence intensity distribution of these GPI-linked molecules at the surface of a mammalian cell, a Chinese hamster ovary cell in this case. And if you notice, the distribution in most parts looks quite uniform and flat. There are some regions which correspond to microvilli that have slightly higher intensity than the surroundings. And there are other regions which correspond to edges of cells which look rather undifferentiated. But if we now switch our gaze to the FRET signal, and here the code is red for very low FRET and blue for very high FRET. that is corresponding to high level of anisotropy and low level of anisotropy. The low level of anisotropy, which is the blue signals, indicate regions where there is enormous amount of fret going on, and the high level of anisotropy indicates regions where very low levels of fret is taking place. And what you immediately see is that there are different shades of fret distributed throughout the cell surface. There is a region of low fret and a region of high fret, which correspond to regions where these clusters of these GPI-anchored proteins are enriched or depleted. If we look more closely, for example, at the microvilli, we find that the microvilli are regions where the clusters of the GPI-anchored proteins seem to be specifically depleted. The same seems to be the case where at tips of lamellipodia. So at tips of lamellipodia, we find that these GPI-anchored proteins also look quite heavily depleted. So these are edges of cells where the cells begin to extend protruding actin-driven membranes, where at the very tips, you see a very high anisotropy that corresponds to the exclusion of these nanoclusters. In fact, in living cells, when we observe cells performing their natural ruffling functions, we find that the cells seem to have seem to be dynamically excluding the clusters from the edges that are ruffling. In fact, ruffling cells, if you look at the arrows when they in the movie, you find that the arrows indicate moments in time when in ruffling cells the ruffles have extended out and the GPI-anchored protein clusters, as detected by the fret signals, are completely depleted from these ruffling edges. Other regions in the cells are where the nanoclusters of these molecules are enriched, and they seem to be enriched specifically in the flat regions of cells, or in regions just behind the tips of lamellipodia, the lamellum, where the distribution of actin is likely to be very different from the tip of the lamellum, of the lamellipodia, or in fact even microvilli. So it appears from these sorts of observations that the GPI-anchored protein nanoclusters are in many cellular environments correlating with the underlying cytoskeletal matrix. If we just look at a flat-scape of a cell, where the membrane is relatively flat at least to the eye of the microscope, we find that there are a patchwork of domains where GPI-anchored proteins are extremely enriched, these very deep blue regions, and GPI-anchored proteins are not as enriched in these slightly yellow regions. But these patchworks are regions which are showing a kind of variegated pattern, which is, when analyzed by some statistical means, shows a completely non-random segregation of clusters. So in other words, it appears that the membrane is not a homogeneous, well-mixed membrane where clusters are present sprinkled uniformly over the surface of the membrane, but in fact there are regions where clusters are selectively concentrated, and they're selectively concentrated or excluded from regions of membrane that seem to have distinct underlying cytoskeleton. Experiments that we have begun to do suggest that the localization of these nanoclusters correlate with specific types of the cytoskeletal organization, specifically the actin organization. in fact perturbing the actin-cytoskeleton using drugs that can impair the polymerization or prevent the depolymerization of actin, or dramatically affect the distribution of these nanoclusters in cell membranes. So it seems as if that we may have something that motivates the formation of these regions in cell membranes where nanoclusters are concentrated. In fact, the regions where nanoclusters are concentrated correlate with regions of actin, where the actin-cytoskeleton is likely to be moving in a arranged in a horizontal fashion beneath the plasma membrane. And regions where these clusters are excluded seem to be correlating with regions where the cytoskeleton is effectively organized vertical to the plane of the membrane. And this may be, in some ways, a mechanism that the cell uses to regulate the local composition of these clusters in different regions of the cell membrane. And then it is quite likely that the sorting and signaling functions that we have ascribed to different types of membrane domains could be realized through this kind of organization of membrane components in the cell. The experiments that I have just shown you are, of course, extremely new and perhaps right, almost hot off the bench, but they are not unsupported by other literature that has been accruing over the past several years, specifically from work from Ken Jacobson's group, Mike Sheets' group, and Aki Kusumi's group in Japan, where they have argued now that the membrane of a living cell is in fact a supported bilayer. It's sitting on top of a cytoskeleton. And the cytoskeleton, in fact, not only, as I indicated to you, influences the local distribution of these nanoclusters in the membrane, it also influences the mobility of molecules on the cell surface. So when Aki Kusumi looks at the movement of a single particle tagged to a fluorophore on the cell membrane, or tagged to a gold particle, if he looks at a lipid or if he looks at a membrane protein, they exhibit sort of a rather characteristic pattern of diffusion when observed at a relatively slow frame rate. When Aki Kusumi looks at the membrane at an extremely fast frame rate, the picture that they observe is quite different. It seems that the particles that move spend times in small confined regions in the cell membrane and then jump to other regions in the cell membrane as if they were exhibiting some sort of a hop diffusion mechanism. And this type of characteristic is only observed when the frame rate for observation is in fact 40,000 times per second. Achievements that are at the moment at the edge of technology today. These regions can be mapped out, and when they are identified by computer programs that can detect breaks in the diffusional characteristics within different regions, or can identify these little domains, they show a fairly interesting pattern of adjacent regions that are organized in some fashion. And when again Aki Kusumi's group looks at the underside of the plasma membrane, very close to the lipid bilayer, they find that the cytoskeleton seems to be also arranged in a pattern that corresponds to these domains in which these lipids seem to be confined or these proteins seem to be confined in diffusional during their diffusional process. So putting these different observations together, it seems as if the membrane of a living cell is not simply the lipid bilayer situated on its own, but it's in fact a bilayer that's in close conversation with the underlying cytoskeleton. And that conversation seems to be generating new structures, rafts, if you want to call them that, or structures where molecules can be confined by the scaffolding of the cytoskeleton for periods of time necessary for engaging signaling output. And that, we believe that those kinds of organizational forms can also generate budding pits at the cell surface. Of course, these are speculations, and I think it's important to be able to clearly state what we know and what is a proposal. So what we know is that we can see small clusters of these lipid-anchored proteins in membranes, whose distribution and whose existence as well seems to be very dependent on the presence of the specific organization of the underlying cortical actin. And that is, it appears, a two-way conversation between the lipids in the bilayer and the cortical cytoskeleton. And this obviously implies that the membrane of a living cell is not a passive fluid mosaic model, but in fact it's an extremely active system, which is I guess a walking, talking composite between the bilayer and the underlying cortical cytoskeleton, which is associated with. And there are multiple things going on in the membrane that could influence the local organization of components. There's transbilayer flip-flop mechanisms which can change the local composition of lipids. There are protein-protein interactions whose aggregation can again change the nature of the lipids surrounding these protein-protein aggregates. There could be synapses in the membrane which could bring other cells close to the membrane, which again can influence the local lipid composition. There are, as I think, determining cytoskeletal interactions, cytoplasmic proteins like negatively charged molecules like Marx proteins, which also can influence the local lipid composition. And finally, the well-characterized clathrin and caveoli coated regions of the membrane, I think, represent one example of how proteins, in fact, and their dynamics can influence the local structure and composition in cell membranes. So I think together, putting many of the pieces of this puzzle, where we have a bilayer consisting of a variety of different membrane components, it seems that the cell is using special organizing principles from these active polymers of the cytoskeleton to locally control the nature, composition, properties of the plasma membrane in a way that I think is going to keep us busy in trying to understand for many decades to come. But I think this is just the beginning of a new way of looking at the cell membrane. And for all the young researchers out there, I think this is a very important area of research, because although there has been work in this area for such a long time, I think we know very little about how the local composition of the cell membrane is regulated. And of course, what I've described to you today is in fact a proposal. And like many, these proposals are open for debate, challenge, and investigation. And I think this investigation has to be a multidisciplinary one, where we are using tools from every resource possible. We're using tools of physics, of the latest physics, of the physics of active systems, chemistry, where we try and understand how the chemistry of the Membrane is coupling to the activity of components in the cell, and also exploring all the tools that the biological system allows us, the tools of genetics and imaging, to bring together and to bring to bear on this fundamental problem of how membranes of living cells are organized. And I'd like to invite all the young researchers out there to this open arena, which I think is just the start of a new adventure in cell biology. Thank you. Yeah.